In this video, we are going to review the structure of DNA. So a lot of this we learned in our first unit when we looked at nucleic acids, specifically DNA. So recall that DNA is made up of nucleotides, and each nucleotide is made up of three things. So let's remind ourselves what that is. So first of all, we have our deoxyribose sugar. So this is deoxyribose. So this is a ribose sugar in that it has 5 carbons, but it's deoxy because it is missing the oxygen here, which the ribose sugar that RNA has would have an oxygen here. Now, we also need to know how to number the carbons, which we had a little bit of practice with in the first unit, but let's remind ourselves. So we are always going to start looking at the oxygen here. So off of that oxygen, the first carbon here is carbon 1, and then we just go clockwise. We have carbon 2, carbon 3, carbon 4, and carbon 5. So carbon 5 is attached to a phosphate group. And these are very easy to identify because we have a phosphorus with a whole bunch of oxygens and negatives and double bonds and just a whole very identifiable group of atoms. And then off of carbon 1, we have 1 of 4 nitrogenous bases. And this shown here is adenine. And remember, we have adenine, thymine, guanine, and cytosine. So these are all the nitrogenous bases found in DNA. In RNA, it's very similar. Recall that RNA, again, is going to have the oxygen here. It's going to have a ribose sugar. But instead of thymine as its nitrogenous base option, it would be uracil. So RNA would have adenine, uracil, guanine, and cytosine. So recall, there's specific bonding of DNA, and the bonds that attach the nucleotides together are phosphodiester bonds. And that's where we have the phosphate group attaching to the next sugar of the other nucleotide. So it's joining the sugar and the phosphate group together. This is going to create a sugar-phosphate backbone. Remember, the outside of what we consider the ladder is the sugar-phosphate backbone. What connects the bases to the sugar is called a glycosidic bond. And remember, any kind of glycosidic bond is a bond that attaches a sugar to another group that may or may not be another carbohydrate. So in Unit 1, we looked mostly at carbohydrates bonding to each other, but a glycosidic bond can be any kind of carbohydrate binding to something else. So looking at base pairing, we have adenine is always going to bond with thymine. Guanine always bonds with cytosine. And you can remember apples and trees, cars and garages. So we have adenine and guanine. These are both similar in the sense that they both have 2 ring structures. And they are called purines. And remember I said we have a larger structure but a shorter name compared to pyrimidines, which are thymine and cytosine, which only have a single ring structure. And again, longer name, shorter structure. So adenine always pairs with thymine and guanine always pairs with cytosine. And these are held together through hydrogen bonds. So let's look at each pair. So if we have adenine binding with thymine, we see 2 hydrogen bonds here. So adenine and thymine will have 2 hydrogen bonds. Whereas we have cytosine and guanine, and they have 3 hydrogen bonds. So that's one way to see the difference in the structure, and it's going to become very important in the behavior of DNA. So Chargaff's rule is that every adenine is going to have the same amount, for the most part, as thymines. And you're going to have the same amount of guanines as you do cytosines. And that's because for every A, it's going to be pairing with a T. So if you have 50 adenines, you should have 50 thymines because they're paired up. Similarly, if you have 100 purines, you should have 100 guanines, you should have 100 cytosines. So those should always be equal. A's should be equal to T's, and G's should be equal to C's. So let's look at some percentages of bases in different organisms. So if we look at Streptococcus, you have almost the same amount of A's and T's and essentially almost the same amount of G's and C's. Similarly for yeast, we have about the same amount for A's and T's, similarly for G's and C's. Same is true for herring, 27-ish and 22. And for humans, you're having about, that's Hannah. This is her trick, which nobody else in the house can do. She has between around 30 A's and T's and less G's and C's, but the same amount of G's and C's. So if you recall from our DNA lesson in Unit 1, DNA is going to run in anti-parallel directions. So that means if one DNA is going in this direction, the other one is going in the opposite direction. And we see this if we look at this strand here. If we look at the O's of the sugars, so the oxygen of the sugar, these ones are, if you will, facing upwards. If we look at the other strand, the O's are upside down. So these sugars are upside down and they're moving in the opposite direction. So those are upside down. And because of this directionality, we do have naming. We have a 5' and a 3' end. If you remember, the 5' end is where we have the phosphate group. Remember, 5-phosphate. So we have that f sound. So the 5' end of the DNA strand has the phosphate. The 3' end ends with the sugar. So the 3' end is always going to have the sugar. And if we see here, the same is true. We have the sugar at this end, and we have the phosphate at this end, at the 5' end. So again, we see the 5' end on this strand and the 5' end on that strand where the phosphate groups are. And then similarly, we have the 3' end on both strands where the sugars are. And again, these are arranged as a double helix, if you remember this. So it's not just a ladder, but we consider this a twisted ladder. And we see there's one strand, one sugar phosphate backbone, and then we have the other sugar phosphate backbone on the outside. And along the middle, we have our nitrogenous bases. And this would be a 3D structure showing all of the elements involved with our nucleotides, again, our nitrogenous bases in the middle, and we have our sugar phosphate backbone along the back. Now, why does this happen? We talked about this in the first unit. Primarily, it is because the nitrogenous bases are hydrophobic. So they are going to be trying to get away from the aqueous environment of the cell, whereas the sugar phosphate backbone is hydrophilic, so it is loving the aqueous environment. There is also the just the basic structure and size of the nucleotides. As they stack, they're going to naturally create a curving pattern. And there is a link in LMS in content for that animates that beautifully for you to look at. So Since we know that A's pair with T's and G's pair with C, if you are given one sequence of DNA, you could provide the complementary sequence strand. So for example, we have our 5' end and our 3' end of a given sequence, and you would be able to provide its complementary strand. So you always start by writing the 3' because this is the 5', so it's going to be the 3' end of the complementary strand. And since this is a 3' end of the given strand, this is going to be the 5' end of the complementary strand. And since we know A pairs with T, the first nucleotide would be T. And then T, we know the complementary base would be A, and so on and so forth. So G is going to pair with C, C will pair with G, A will pair with T, then we have A, C, T, A, T, T, C. So let's look at this diagram to be able to provide some of the definitions that are in your note. So we see we have here our double helix, and we know we have our sugar phosphate backbone, and we have our complementary nitrogenous bases. As we follow along, we have these histones, which are proteins that the DNA wraps around. And this is so that the DNA can, A, fit in the nucleus and even fit within the cell, and to protect the DNA from anything that might deteriorate or degrade the DNA. So it needs to be protected. And we also don't want just random breaks from happening in the DNA. So wrapping around the proteins allows the DNA to be super structured and condensed and to protect it from any kind of damage. And so we have these histones that the DNA is going to wrap around. And these are called, again, we have histones that are just the proteins that the DNA wraps around. This structure that with the DNA and the proteins together, so that structure here, this is called a nucleosome. So again, it's just the same thing, different terminology. Protein itself are histones. The DNA wrapped around the protein is called a histone. And then we have, it's called a nucleosome. And then we are going to have the nucleosomes wrapping around into a spiral-like structure. And again, this just helps to condense the DNA. So let's look specifically at the terminology. Again, histones are structural proteins that the DNA is going to wrap around. Nucleosome is the section of DNA that is wrapped around the histones. So again, histones are the protein, and then the DNA wrapped around the proteins are called a nucleosome. Chromatin is the entire section of DNA that has been wrapped around the proteins. So it's the DNA and histones. So this would be a chromatin, this whole thing showing here. Furthermore, chromatid is what we would call a single chromosome that is wrapped around the proteins, so it's condensed. So chromatid is essentially the same thing as chromosome, except it has been wrapped around protein. When DNA replicates, and this would be during the S phase of interphase, remember that? We have two sister chromatids, and these would be identical chromosomes. So we would have sister chromatids. So this is a chromatid, and this is a chromatid. They would also be called essentially chromosomes. We have the sister chromatids that are connected at a specific sequence of DNA, and that is called a centromere. And then lastly, the chromosome is the highly condensed chromatin. So chromatin would be loosely wrapped, like here. And then a chromosome is where it's going to be super, super condensed. The last definition the last slide that we have, is a gene. And so this would be a section of DNA that codes for a specific protein. And if you remember from grade 11, you looked at a bunch of different diseases related to mutations in different genes. And you may be familiar with the fact that you have a lot of different genes on a single chromosome. And those genes aren't necessarily related to each other. And you can have genes that are related to each other on different chromosomes. So it's all very interesting when you look at the genetics and genes of different organisms. And that is it for our introductory lesson on genetics. We are going to look at the history of DNA research, very exciting. And then we're going to look more at how DNA replicates and how proteins are made from DNA. So that's the majority of the rest of this unit.